Phylogenetic trees and cladograms give us a sense of the order of evolution. They show us a diagram indicating how groups split and approximately the order of that splitting and of those traits arising. But they don't actually tell you about the actual time that things happened. They don't tell you how long it took to get from one trait to another, for example. And so it's important to realize that on a cladogram, when we're drawing like this, for example, we tend to make our drawings just kind of evenly patterned when we're not using time. And so that would suggest that it took equal time to get from each different trait transition. But again, that doesn't really mean anything. I've just drawn my cladogram to look like that. The real sequence of things might have been that one thing took a really short amount of time and another trait took a really long time to evolve. So this might be the actual true or relative order of events, but we tend to draw it this way just to make room for drawing and things like that. Okay, so again, the cladogram or phylogenetic tree itself doesn't necessarily reflect time. If we want to have time reflected in our phylogenetic tree, we have to measure time in a different way and then superimpose it. So many of our phylogenetic trees will reflect time by having a time scale over next to them. So if a cladogram or a phylogenetic tree is reflecting real time, you should see a time scale over to one side of it to show how long things took. And so how do we get that time scale? The time scale comes from fossils. So we can use fossils to come up with, okay, this organism was around here, this organism was around here, this organism was back here. So you can use fossils that way if you have a fossil record for that. Now remember, most of the time we don't have a complete fossil record. So most of the time we can't do that with fossils. So a lot of times we rely on something called molecular clocks. So a molecular clock is going to use changes in the DNA to estimate time. Okay, so a molecular clock is a time estimate. So it's got a couple of assumptions behind molecular clocks. So the first is that most mutations are neutral. Okay, so the majority of mutations that we get are just going to be silent in our DNA. It's because they're not in places where they're affecting our genes or affecting our traits. So favorable mutations, if those are beneficial, they're rare, but they will increase in the population. And negative mutations, again, they are not going to happen as much, but they're just going to be eliminated. Okay. Neutral mutations, again, they're happening all the time, but they're not influenced by natural selection. Because they're neutral, they're just going to build up in our DNA, and they will neither increase in frequency nor decrease in frequency. So neutral mutations are occurring at a constant rate generally, and so they can therefore measure time. So the amount of neutral mutations that we have is a readout for time. Okay, so we get increased neutral mutations as we have time progressing. Okay, so if I look at the difference in those mutations between two species, so if I compare species A to species B, the higher the number of differences, the more time since they've diverged from each other. So the more time since they diverged from their ancestor. So longer periods of time since divergence equals a greater number of neutral mutations. The opposite is also true. Shorter periods of time, less neutral mutation differences. So is this a perfect estimate? No. We know that not all organisms acquire new mutations at the same rate. So organisms have different rates at which they get mutations. A lot of this has to do with the difference in generation time. So again, bacteria are going to reproduce faster and acquire these mutations differently than us. And so as much as possible, a molecular clock is also matched against the fossils that we do have. So we take the molecular data, we compare it to the fossil record, and we put those two things together in order to make a molecular clock for a species. And so that's going to kind of look like this, right? So the nucleotide differences is on the y-axis, and then the time since two species diverged is on the x-axis. So the greater the difference, the more time. And so if I have a value, I can say, okay, these two monkeys have this much percent difference, and then I go across in my graph, and I say, okay, that suggests that it's this many years since those two species diverged from each other. Okay, so these might be my actual data points where I have like a fossils and also percent differences. And so the line gets filled in to approximately fit those data points. And again, I can use it to predict for other organisms. So just to check in on here, when comparing the sequence of a homologous gene in 4 different species, one pair shows a 1% difference, the other pair shows a 3% difference. Which pair diverged longer ago from their common ancestor? Well, remember, the more differences equals the longer time ago since divergence. So the 3% difference group would have diverged a longer time ago. So this is, again, an example of a time scale that has put next to a cladogram. So this is a cladogram showing some primate phylogeny, and then we've used a molecular clock and the fossil record to put a time scale next to it. And so notice that the branch points are shorter or longer. So this is a longer one than this one. And so that we're able to do that because we've superimposed a time scale. But again, when we make cladograms, especially in class or as an assignment, we're generally not going to have a time scale. So we're just evenly distributing. It's not suggesting time. It takes this extra piece of research to make the time scale in order to make our cladograms reflect true time. The cladogram itself just tells us the order of events.